My dear student, today let us see the digestion and absorption of fat. The last class we have seen about carbohydrates and proteins. The diet contains fats of vegetable origin and also fat origin. Mostly it is in the form of triglycerides. Small amount is present as phospholipids, cholesterol and cholesterol esters. The digestion and absorption of fat is more complicated and time-consuming process than carbohydrates and proteins. What is the reason? Why it is complicated? Why does it take more time for the digestion and absorption? Yes, it is water insoluble. If you take some mutton and wash it in a bucket of water, what will happen? The fats are floating on the surface of the water. Like that, the fat in the intestinal lumen are floating and they are facing difficulties to approach or contact the epithelial cells, intestinal epithelial cells for absorption. And also the lipase, which are facing difficulties to act on the fat to digest it. Therefore, it takes more time and it is complicated. The last class we have seen the bond present in the carbohydrate as well as in the protein, that you know very well. It is, the bond is, the carbohydrate is linear one, it has 1,4 alpha glycosidic linkage. At the branching it has the 1,6 glycosidic linkage. The only digestive enzyme to hydrolyze the bond at the branching, I mean 1,6 glycosidic linkage, is alpha limit dextrinase. What is the bond in the protein that is to be hydrolyzed for digestion is peptide bond. Like that, here, I mean the fat, already I told you most of the fats are in the form of triglycerides. Therefore, the discussion regarding the digestion and absorption is mostly centered around the triglycerides. This is the glycerol backbone consisting of 3 fatty acids. Therefore, it is called triglycerides. The bond between the glycerol backbone and the fatty acid chain, side chain, is ester bond, ester linkage. This should be hydrolyzed. This should be broken. This is the ester bond. Oxygen, double bond with carbon, and this carbon is binding with the side chain. Here also with the binding with the oxygen. This is the ester bond. Our discussion regarding digestion and absorption of fat is mainly centered around the triglycerides. To digest the triglyceride, what bond to be broken? Just now I told you, the ester bond should be cleaved. The action of gastric lipase is insignificant, because digestion of all the dietary fat occurs in the small intestine with the help of pancreatic lipase. What are the lipolytic enzymes present in the pancreatic juice? And what is the digestive function of the lipase? The pancreatic juice, we have 3 lipolytic enzymes. They are very important. pancreatic lipase, cholesterol esterase, and phospholipase A2. What is the function of pancreatic lipase? Yes, it is acting on the triglycerides and produce 2 fatty acids and 1 monoglyceride. Cholesterol esterase acts on cholesterol ester and produce 1 cholesterol molecule and 1 fatty acid chain. Whereas phospholipase hydrolyzes the phospholipids. Now let us see about the emulsification. The first step in fat digestion is emulsification. The process by which large fat globule is converted into fine droplet is known as emulsification. How does it happen? Yes, the bile salt reduces the surface tension of fats. So, during segmentation movement of small intestine, large fat globules are splitted into fine droplets of 1 micrometer diameter. Thus, emulsification exposes large surface area of triglyceride to the action of pancreatic lipase. After emulsification, bile salts separate the fat droplets like this. This is the large globule before the emulsification. After emulsification by the bile salts, this large globule is split into fine droplets. This should be removed immediately from the place. Then only further emulsification will be taken. This is done by the bile salts. The bile salts are arranged on the surface of the globule like this. I mean, heads are on the surface of the globule, tails are interior of the globule. So, this is the bile salt. Polar head, bile salt, this is the tail. This is the, water insoluble, heads are water soluble. Polar head, it's a non-polar tail. Since the surface of the droplet is covered by the heads of the bile salts, the whole droplet is converted into water soluble one. Therefore, they can move easily to any place in the intestinal lumen. Now it is converted into water soluble. The whole is converted into water soluble. Emulsification actually helps for hydrolysis. But what happened? The bile salt molecules present in the bile salt molecule present the membrane around fat droplet inhibits the activity of lipase. Bile salt membrane, this one, the bile salt membrane around the droplet inhibits the activity of lipase. Therefore, those those bile salts should be displaced. Then only pancreatic lipase can act on it to digest the ester bond, to release the fatty acid. Therefore, co-lipase, a protein present in the pancreatic juice of molecular weight 10,000, displaces the bile salt and allows lipase act on the droplet. So, what is the function of the co-lipase? It displaces the bile salt from the droplet for the action of pancreatic lipase on the droplet for digestion. Now let us see the absorption. Before studying about the absorption, we should know the formation of micelle. Lipids are water insoluble, so they cannot move easily in the intestinal lumen to contact epithelial cells for absorption. Therefore, micelle helps for the transport of water insoluble fats. Bile salts aggregate in the form of small globules with their tails in the center and heads fanning out to cover the surface of the globule. The bile salts are arranged like this. The water soluble heads are on the surface of the globule and tails are towards the center of the globule. Therefore, the whole globule is converted into water soluble. But what is at the center of the globule? The center is tail. It is water insoluble, but it is lipid soluble. Therefore, what will happen after the digestion? The fatty acid, after the digestion also, the product of digestion are still water insoluble. They are easily binding with the micelle, bile salt micelle, because at the center the tails are lipid soluble. Lipid soluble. Therefore, the lipids are binding at the center of the bile salt micelle. Now it is converted into mixed micelle. Mixed micelle. Therefore, micelle helps for the transport of water insoluble fats. Bile salts aggregate in the form of small globules with their tails in the center and heads fanning out to cover the surface of the globule. Each micelle is composed of 30 molecules of bile salts and 2.5 nanometer diameter in size. The products of digestion are still water insoluble. Monoglycerides and fatty acids, they are binding at the center of the micelle formed by bile salt. The center of the bile salt micelle is lipid soluble, so they are binding easily. But the whole surface of the micelle is water soluble. Therefore, micelle transport the water insoluble triglyceride and fatty acid towards the epithelial cell for absorption. Monoglycerides and fatty acid diffuse freely into the epithelial cell. Bile salts are released into the lumen to repeat the process. The hydrolysis of fat is highly reversible. The accumulation of product of digestion will stop the process of fat digestion. So the products of digestion should be removed from the intestinal lumen. The micelle is removing the triglycerides and fatty acids and allowing for further digestion of fats. The bile salts must be present in certain minimum concentration for the formation of micelle. I mean bile salt micelle. This is known as critical micelle concentration. Cholesterol esters and phospholipids are also digested and absorbed as triglycerides. What is happening now? The product of digestion of fat enter into the epithelial cell. What is happening to the products of fat digestion after entering into the epithelial cells? To complete the absorption, they should enter into the blood circulation. How does it enter into the blood circulation? Small fatty acid consists less than 10 carbon atoms, cross the basal border by simple diffusion and enter the portal blood. But fatty acids with larger chains are esterified into triglycerides in the intestinal epithelium. Cholesterol also esterified into cholesterol esters. These esterifications are taking place in the endoplasmic reticulum of the epithelial cell. The absorbed phospholipids form a membrane around small fat globules, which is consisting of triglycerides and cholesterol esters. A small amount of beta lipoproteins also incorporated into the membrane of the fat globule. This globule of 60 to 75 nanometer diameter is known as chylomicron. What is chylomicron? What is chylomicron? Yes, chylomicron is a fat globule which is consisting of, at the center, which is consisting of triglycerides and cholesterol esters. On the surface of the globule, which is consisting of head of the phospholipids. On the surface, it has beta lipoproteins also. This globule is known as chylomicron. Chylomicrons are too large to diffuse through basal border of the epithelial cell. So they are released into the lacteals and ultimately enter the blood circulation through thoracic duct. The absorption of dietary fat is completed in the jejunum. Bile salts are not absorbed along with the products of fat digestion. Then what happened to the bile salts? They are absorbed in the terminal part of ileum. Bile salts are made up of bile acids. Conjugated bile acids seem to be absorbed by secondary active transport utilizing sodium carrier protein. Non-conjugated bile acids seem to be absorbed by simple diffusion. If digestion and absorption is imperfect, then it leads to bulky, greasy stools, foul smelling, and fecal fat contents up to 40 to 50 gram per day. So this condition is known as steatorrhea. For the digestion and absorption of the fat, what is necessary? Bile salt and pancreatic lipase. You know very well bile salt is from bile, that is liver, and pancreatic lipase, it's obvious, it's from the pancreas. If these two, pancreas or liver, liver is affected means, then the lipid digestion will be affected and it leads to steatorrhea. Steatorrhea is a condition in which digestion and absorption is imperfect. This imperfect digestion and absorption of fat leads to bulky, greasy stools, foul smelling, and fecal fat contents increased up to 40 to 50 gram per day. This is steatorrhea. Now we have seen digestion and absorption of carbohydrate, protein, and fats. In the carbohydrates, If it is linear one, 1,4 alpha glycosidic linkage should be broken. In the case of protein, the peptide bond should be broken to digest the protein. Like that, in the case of fat, especially triglycerides, the ester bond should be hydrolyzed. 1,6 alpha glycosidic linkage will be hydrolyzed only by the alpha limit dextrinase, the only enzyme present in the GIT to break the bond. Break the bond. 1,6 alpha glycosidic linkage is alpha limit dextrinase. Cellulose cannot be digested in the human GIT because it has the beta glycosidic linkage, not alpha. Therefore, there is no enzyme acting on the beta glycosidic linkage. Therefore, cellulose cannot be digested in the GIT. What is digestion? What is digestion? Digestion is a process by which large complex molecule is converted into small, simple, absorbable molecule. That is known as digestion. What is absorption? Yes, absorption is a process by which the small absorbable molecule entering into the blood circulation. This is known as absorption. Okay. Thank you.